today we will start chapter 7. Chapter 7 title is Cell Communication and Multicellularity. Chapter 7 is composed of 5 subjects starting from the signal and receptor, signal transduction, and also cell changes in response to cell signal, and also we have to know the way how to adjacent cells communicate each other. So first of all, I will explain the first subject, cell detect a variety of signal. Okay, so first in this slide, I will explain the cell signaling on the local level, and the next slide, I will explain the cell signaling from a distance. Okay, first of all, Marin and Rhea, don't talk to each other, okay? First of all, I will explain cell signaling on the local level. There are 3 kinds of signal. First of all, autocrine signal. Second one is juxtacrine signal. Final one is paracrine signal. Okay. So this cell is the secreting cell, and this cell is the target cell, and this cell is also target cell. Okay, let me start from the autocrine signal. Okay, so this is recorded as the autocrine signal. So this secreting cell secrete signals, and also this signal is bind to the receptor on the same cell that secrete them. So this one we call it as autocrine signal. So bind to the receptor on the same cell that secrete them. Okay, next one is the juxtacrine signal. Okay, so this is the secreting cell, and this is the targeting cell. So this signal is bind to adjacent cell in contact. So just this signal, okay, the target cell has the receptor, and this signal is bind to the receptor in adjacent cell in contact. This is recorded as juxtacrine signal. Final one is that this one is recorded as paracrine. So this secreting cell secrete signals. This target cell is adjacent cell in contact. How about this cell? This paracrine signal is bind to the receptor on the nearby, not contact, nearby cells. So this is recorded as the cell signaling on the local level, autocrine, juxtacrine, and paracrine. How about cell signaling from a distance? Okay, so let me start from the hormone. Hormone is signals. So secreting cell secrete hormone, and the targeting cell here and here. They are far away from this target cell is far away from secreting cell. So this signal is traveled through circulatory system of animal. So for example, in the animal, we have the circulatory system. So this signal is traveled through the circulatory system of animal, or for example, in the plant, this signal is traveled through the vascular system of the plant. So this one we call it as a hormone. Okay, so our body has, for example, hypothalamus secrete TRH and DRH, something like that. Okay, thyroid secrete hormone, we call it a PTH, like this. Okay, so in the cells from a distance, this circulating signal is transported by circulatory system and bind to the receptor on a distant cell. So this one we call it as the cell signaling from a distance. In this case, is hormone is involved in this signal. Okay. Okay. And also, let me explain the signal transduction pathway, starting from the signal, receptor, and responser. Okay. Okay. Let me explain the. Okay, in the midterm exam, I can ask you like this. Explain about the signal transduction pathway. First one is signal. This signal is that secreting cell secrete this signal, and the receptor. And the receptor is the target cell have a specific receptor that can detect the signal. That final one is the responser. So this is the inactive transduction molecule. So apt signal bind to the receptor on a target cell. So this can activate this inactive signal transduction molecules. So these inactive signal transduction molecules become the activated, like this. And also this activated molecule can activate second signal, like this. Okay? And there are two kinds of the responser. First one is short term, and second one is long term. For example, this signal molecule can be activated. Okay, at this time, this effect is maintained only short time. However, when this signal signal transduction molecule is activated, when this signal transduction molecule is transported to the nucleus, at that when it functions as this signal transduction molecule bind to the DNA, it can change DNA transcription level. When this happens, we call it as long-term changes. Okay, so short-term changes only involved in enzyme activation, some kind of signal transduction molecule activation. However, when activated signal transduction molecule is transported to nucleus, at that when it binds to the DNA, it can change the DNA transcription level. When this is happen, we call it as a long-term changes. Okay. Okay, and let me go to the second subject, the receptors. Receptors bind to the signal to initiate a cellular response. Okay, so receptor, so this is the receptor on the membrane. Okay, the receptor have specific binding site for signal. Okay, this receptor has the binding site here. So signal can bind to this location. So signals fit into three-dimensional site on its receptor. So this is fit into this region. After that, it can initiate a cellular response. Okay. Okay. Second subject, we learn about the receptor. Okay. Receptor have a specific binding site for signal. Okay. Signal is directly bind to this location. After that, it can initiate a cellular response. Okay. Also, there are signal can bind to this receptor, and also other chemicals that resemble ligand, that resemble signal, can also bind to the receptor. Okay, we call it as an agonist, and also the other one we call it as an antagonist. Okay, so usually, this is a receptor. So usually the signal or the ligand can bind to this receptor. After that, signal can be activated. Okay, let me start from the agonist. Agonist is resemble the ligand. Agonist resemble this ligand, so it also can bind to the receptor. Okay, sometimes it can induce 100% activation. Sometimes it can induce less than 100% activation. Okay, so agonist, the important thing is agonist is resemble this ligand or this signal, and also can bind to the receptor region. After that, it can activate. It can induce the full activation or less than 100% activation. Okay, let me start from another example, antagonist. Okay, so usually the hormone and the neurotransmitter function as signal. It always binds to the receptor. That it can induce full activation. However, antagonists also bind to the receptor. After antagonists bind to the receptor, it prevents this ligand to bind to here. So after antagonists bind to the receptor, it prevents the ligand from binding. After that, there is no activation. Okay. So in the next slide, I will show you one example of the antagonist. Okay. So caffeine, when we drink the coffee, coffee has caffeine. Caffeine function as an antagonist. I will show you the detailed information in the next slide. Okay. For example, so I will explain some example of antagonist caffeine. Okay, so let me start from the one signal. So adenosine function as signal or the hormone. So usually this adenosine bind to the adenosine 2A receptor. At that, it can initiate several signal transduction pathways like this. Okay, at that, it can reduce its brain activity. So when adenosine bind to the adenosine 2A receptor, it can reduce brain activity. It means that people fall asleep. Okay, when adenosine bind to the adenosine 2A receptor, and sometimes I found that some people fall asleep. So at that time, adenosine bind to the adenosine 2A receptor. At that, 조아연 becomes 멍, like this. Okay. However, caffeine functions as antagonist. So because the caffeine's molecular structure is nearly similar to the adenosine. Okay, they are nearly similar to each other. So caffeine also can bind to the adenosine 2A receptor. At that preventing adenosine binding, okay, it prevents adenosine binding, thereby increasing brain activity. That's why caffeine has function as awakening. Whenever we drink coffee, we became awakening, okay? Because our brain activity is increased. Okay, this is one example of antagonist, okay? Okay. Now I will explain the receptor, detailed information of receptor. Okay, receptor has the two kinds. So we already learned that they have the membrane receptor, and also have another kind is intracellular receptor. In chapter 6.3, chapter 6, we learned that diffusion. Okay, there are two kinds of diffusion. At that time, we learned that. Okay, small and uncharged molecule can pass through the hydrophobic lipid bilayer. Okay. For example, intracellular receptor. So inside the cell there is a receptor. So when small non-polar and small uncharged molecule can pass through the lipid bilayer. Okay, and at that it can bind to the intracellular receptor. How about the large molecule? So large glucose and amino acid and other the ion Ca2 plus, Na plus, and this kind of ion, large and also polar ligand can bind to the membrane receptor. At that it can initiate the signal transduction pathway. Okay, two kinds of the receptor. Okay, you have to know the difference. Okay. Okay. To solve the midterm exam, you have to know the difference between membrane receptor and intracellular receptor. Okay. Okay. So let me start from the membrane receptor. Okay. In this slide, I will further explain the membrane receptor, and also next slide. Finally, I will explain the intracellular receptor. Okay, let me start from the membrane receptor. There are three kinds of the membrane receptor. First one is the gated ion channel. Second one is protein kinase receptor. Final one is G protein coupled receptor. Okay, let me see the detailed information. Starting from the gated ion channel. Okay, gated ion channel. Okay. Okay, for example, here you will see that. So this is the one example is the acetylcholine receptor. Okay, usually in our neuron, neuron secreted acetylcholine. After that, our muscle has the acetylcholine receptor here, like this. Okay. So this acetylcholine can bind to this receptor. So this is located in the membrane. That's why we call it as membrane receptor. Okay. After acetylcholine bind to the receptor, it can initiate it can initiate changes in shape. At that, it can open the channel. Okay, so inside of the channel are negatively charged like this. That's why the positively charged Na plus can pass through this channel. Okay, so finally the channel open, allowing ion, for example Na plus, this is charged molecule, can flow into the cells like this. Okay, so acetylcholine bind to the here. After that, there was of the channel protein. After that, the ion, especially the Na plus, can flow into the cells like this. Okay, so this is the gated ion channel. Okay. Another second species is protein kinase receptor. Okay, protein kinase receptor. So signal, for example, insulin can function as signal. Okay, signal bind to the receptor. Okay, so after insulin bind to the protein kinase receptor, so after this binding, phosphorylated itself. This binding phosphorylated itself. They have the tail region like this. So this binding can phosphorylate this after signal bind to the receptor. Receptor can phosphorylate itself, after which changes its shape is like this. That this activated receptor can phosphorylate the target cell. At that, it can induce triggering cellular response is like this. So this is protein kinase receptor. Okay. To solve the midterm exam, you have to know the details information. Okay. So I will give you this picture. You have to explain the details procedure. Okay. Okay. The final membrane receptor, we call it as G protein coupled receptor. So this is a receptor, but the receptor also has the G protein. That's why we call it as a G protein coupled receptor. Okay, so usually the signal bind to the receptor. Okay, signal can bind to the receptor. So this is inactive G protein. This binding can activate the G protein. Especially the G protein has alpha, beta, gamma subunit. Especially the alpha subunit has the GTP, but when signal bind to this receptor, so this GDP can become GTP. So at that, it can activate the G protein. So when G protein is activated, one subunit, especially the alpha subunit, can move through the membrane. It can move through the membrane to effector protein. At that, it can activate this effector protein, so it can induce cellular response like this. So let me summarize once again. The final membrane protein, we call it as G protein coupled receptor. So G protein is bind to the receptor. So signal bind to the receptor. Okay, it can activate the G protein. It can activate the G protein. So at that, so one subunit of the G protein move through the membrane. So finally, it binds to the effector protein. At that, effector protein can be activated. At that, it can induce several cellular responses like this. Okay, you understand it? Okay. And now I will explain another intracellular receptor. Okay. Previously, we covered that there are two kinds. One is membrane receptor, usually the large, and also polar ligand can bind to the membrane receptor. So acetylcholine is, for example, acetylcholine is large. This is a large molecule. Okay, and also insulin is also very large molecule, and also polar molecule. And final some kind of hormone is also large and polar molecule. Okay, for example, intracellular receptor usually small and non-polar ligand. Okay, so and usually small and non-polar ligand. Most of the intracellular receptor are transcription factor. They are very small non-polar ligands. Okay, so this is the all of the intracellular receptor are function as the transcription factor. So after it binds to the ligand, so this receptor can move to the nucleus. So it can bind to the DNA, and finally it can alter, change gene expression. So this is recorded as a long-term responder. So in signal transduction pathway, we learn about short-term changes. So when this kind of the signal molecule is activated, this effect only can be maintained a short time. However, for example, some kind of when this receptor is located in intracellular, okay, the hormone is bind to the signal bind to receptor. Sometimes this receptor function as transcription factor, and it can transport it to the nucleus. After finally it can change the transcription. So this is recorded as a long-term changes. Usually the intracellular receptor, most of the intracellular receptor function as transcription factor. After it binds to the ligand, it can move to the nucleus, bind to the DNA, finally change it, alter gene expression. It can induce long-term changes. Did you understand it? Okay. Okay. Let me start from another third subject. Signal transduction spread the responder to signal through the cells. Okay, so we call it as the chain of events or cascade of events. Okay, chain of events means that one thing is happened, after that second thing is happened, after that third thing is happened. So this is chain of events. We call it as a chain of events. So, and also cascade of events. Or this is cascade. In Korean, we call it as a 폭포. Okay. 폭포 starting from the one drop of water. After that, it becomes the cascade like this. Okay. The signal can initiate chain of events, or it can initiate cascade of events like this. Okay, for example, initial signal can bind to the receptor. After that, for example, this is the kinase receptor. After that, this binding can phosphorate itself. It can change its shape. After that, this signal can be amplified like this. That was one signal. But when receptor when activated, it can amplify the signal. It can, okay? So it will become the cascade like this. Starting from the one drop of water, but became the cascade. Okay, starting from one drop of water, but it can be and result in several different responses like this. One A responser, B responser, C, D, E, F like this. Okay, so it means that signal transduction can spread. Okay, can spread through the cell. Because signal can be amplified like this. We call it a chain of events or cascade of events. Okay. Okay. So this is one example of the signal transduction. Okay. So in normal cell. In normal cell, for example, this is Ras protein. So when signal is bind to the when signal bind to the receptor, okay, it can phosphorylate the kinase receptor like this. After that, this kinase receptor can activate the last signal. Okay, we learned that protein kinase receptor. So receptor can phosphorate itself. After that, this receptor can phosphorate target proteins like this. So it can phosphorylate the target last protein. So it can be activated like the LAS and GTP. So it is activated. Okay, but in normal signal transduction pathway, this activate the last signal can be inactivated like this. So activation and inactivation, activation, inactivation. So it happens like this. So it means that it can brief stimulation of cell division. So it results in normal cell division. Okay. However, when last signal is some kind of the function becomes abnormal. So usually in most of cancer cell, last function becomes abnormal. Okay, so when last function becomes abnormal, so when abnormal last becomes activated, so it cannot be inactivated. So it can induce constant stimulation of cell division. At that, it will become the cancer. Okay? So one signal can initiate chain of the cascade of events. At that, it will become the cancer. So this is one example of the signal can initiate a cascade of events. Okay, so in normal condition, last protein can be inactivated, easily can be inactivated. But in cancer condition, this last cannot be inactivated, so signal can be amplified, so it can causing continuous cell division. It will make the cancer. Okay, and also, so previously this is the protein kinase cascade. Okay, so for example, this is protein kinase cascade. Okay, signal can be amplified at each step. So one signal can be amplified, and one signal can be activated, and one signal can be activated. So each time signal becomes 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, like this. So protein kinase cascade means that signal is amplified at each step, like the cascade. So at that, finally, the protein kinase signal can go to the nucleus. Okay, so information that arrived at cell membrane, especially the protein kinase, so this information can go to the nucleus. Finally, it can induce long-term changes. Okay, usually the MET kinase is bind to the DNA, so it can initiate the long-term changes. Okay, so this is the protein kinase cascade. Signal can be activated, amplified at each step. Finally, this signal can be transported to the nucleus. Okay. Okay, so in this slide, I will explain the second messenger. Okay, so at that time, I did not mention about what is second messenger. Now I will explain what is second messenger. There are several kinds of second messenger, CAMP, and also IP3, and also NO. Nitric oxide also can function as the second messenger. Okay, let me see some detailed information, starting from the CAMP. Okay. Okay. So second messenger, we call it as the small molecule. They mediate some step in the cascade. Okay. So second messenger also can amplify the signal. So one example is cyclic AMP. We call it as the CAMP. So actually, this is the ATP. So they have the adenine, and they have the ribose, and they have the 3 phosphate is like this. Okay, this ATP, when 2-phosphate is removed, and so they will become the cyclic AMPs like this. So usually the ATP becomes the CAMP plus 2-phosphate. So at that time, another enzyme's adenine cyclase function. The important thing is this CAMP can function as second messenger. For example, this CAMP can involved in loss of the cellular function. Okay, the CAMP can activate many enzyme targets. Okay, so this is the cascade. Sometimes the CAMPs involved in this step. So it means that it can activate, it can amplify signal. The CAMP can involve in many cellular functions like this, because this messenger can amplify the signal, especially CAMP. Another one is IP3. So this is our lipid bilayer. So we have the PIP2. They have 2 fatty acids, and the one is they have phosphate, and this is the one hat, and this is we call it a PIP2. So some second messenger are formed from membrane phospholipids. PIP2 is membrane phospholipids. Okay, so when this is hydrolysis happens, it will make the 2 components. This head becomes IP3, and this becomes the DAG. So it will become IP3 and DAG. This IP3 and DAG function as the second messenger. Especially this IP3 is function as last of the most of the second messenger is the IP3 like this. Okay, you don't have to memorize the structure because this is biology course. You don't have to memorize course. But they have 3 phosphate like this. So this is why we call it as the IP3. But this one or this one can function as a second messenger. Okay, let me start from the IP3. Okay, so this is the, for example, so in the phospholipids, one phospholipid becomes IP3 and DAG. So when acetylcholine binds to this receptor, okay, it can activate PIP2 hydrolysis. So it means that it will make IP3 is like this. So IP3 can function as a second messenger. The main function of IP3, it can bind to the calcium channel on the ER membrane. After it opens calcium channel, it means that calcium can be transported into the cytoplasm like this. Okay. So it means that IP3 also can function as a second messenger. How about calcium? Calcium is also can function. Calcium ion can function as second messenger. Especially the calcium can stimulate the nitric oxide synthase, so it changes arginine to nitric oxide. Because we are not, so today is the biologicals, you don't have to memorize detail, but L-arginine becomes nitric oxide, it's like this. So it means that calcium ion function as second messenger, so it can amplify the signal. Okay, and also how about nitric oxide? nitric oxide also can function as the second messenger. So nitric oxide can function as second messenger. So we have smooth muscle cell. So usually the nitric oxide can be diffused to smooth muscle cell. So where it can activate it with, so where it can make the cyclic GMP. So GTP becomes cyclic GMP plus 2-phosphate. Okay, so this GMP promote relaxation of smooth muscle cell. At that, it allows more blood flow. Okay, Viagra. Okay, nitric oxide changes GTP and GMP. At that, lots of blood can flow into the blood cell, smooth muscle in blood cell. So this is smooth muscle in blood cell, blood vessel. They have endothelial cell, smooth muscle. It means that it can relax the smooth muscle. After that, it allows more blood flow, so it can induce our erection is like this. Our man has erection is like this. But Viagra does not directly function to nitric oxide, but Viagra can activate this signal, okay, like this. But important thing, so really similar pathways like this. Okay, so today we want to learn that nitric oxide function as second messenger. Okay, so it stimulates cGMP synthesis. So cGMP promote relaxation. It was make relaxation of the smooth muscle cell. So it means that lots of blood can go to the inside. So it will make the penile erection is like this. Okay. The important thing is nitric oxide function as second messenger. Okay, so in this slide we will learn about regulation of signal transduction. So I already mentioned that so in normal LAS function, LAS can be activated. At that, it can be inactivated. Okay, like this. So usually our signal transduction pathway, sometimes, for example, some signal can activate. So it can activate some enzyme, also G protein, or it can make the cyclic AMPs like this. So some signal can lead to the production of enzyme, active G protein, and cyclic AMP, or other enzyme can inactivate this transducer. So it can inactivate this active enzyme, it can inactivate this active G protein, it can inactivate this cyclic AMP. So cyclic AMP becomes AMP is like this. So in our bodies, our signal transduction can be regulated very well. But sometimes something happens, so our signal transduction pathway is not actively regulated, so it will make the cancer is like this. You understand it? Okay. Now I will explain the cell changes in the response to the signal in several ways. Okay. and also enzyme activity changes in response to signal. Okay, here so our cell changes in response to signal. Okay, so when odorant molecule, okay, our nose, okay, at that we have some hypothalamus. Maybe this is located in hypothalamus in the brain. So brain we have the neuron like this. So this odorant molecule can bind to the receptor in the brain. Okay, so usually this brain, okay, this signal can be go to the brain. So this odorant molecule bind to the receptor. So usually this receptor in the brain can activate the G protein. Okay, so this activates G protein. So this activates G protein can activate the effector. The effector name is adenyl cyclohydride. So this adenyl cyclohydride changes ATP to CAMP. So CAMP open the channel. So upon the channel, so some of the ion can be transported inside to the brain. So we thought that this odorant is something like that. Okay. So ion channel. So we have the ion channel, or so some odorant molecule bind to channel. So finally, our ion channel can be opened. So we know that this is the we can know what is this odorant molecule. Okay. Our cell is response to the signal. We respond to signal. At that, some kind of ion can be transported inside to the brain. So brain thinks that this molecule is something like this. Okay? 향수가 왔을 때 우리가 이제 향수를 맡을 수 있는 이유는 오픈 채널이 오픈됐기 때문이죠, 맞죠? Okay. Another thing is that so enzyme activity changes in response to signal. Okay. For example, one inactive protein kinase becomes activated. So, and also this active enzyme, another enzyme was inactivated, but this active enzyme can change its enzyme activity. And also this enzyme also can change another enzyme activity. Finally, this enzyme changes glucogen to glucose 1-phosphate like this. So enzyme activities also can be changed. So this cascade of reaction lead to the altered enzyme activity. Okay? So our cell can respond to signal. So when we respond, we can change the enzyme activities like this. Inactive enzyme becomes active like this. Okay? Finally, we cover adjacent cell in multicellular organism can communicate directly. Okay. Previously, we learned that cell signaling on local level, we have autocrine, paracrine, and juxtacrine, and paracrine. So when also in distant location, we use cell signaling from distance. We can use the hormone. So hormone can travel through circulatory system of animal or vascular system of plants. But sometimes cell can signal each other by directly. They contact each other, but they have some channel like this. Okay. One is in animal cell, we call it as the gap junction. In plant cell, we call it as plasmodesmata. Plasmodesmata. In Korean, we call it as 원형질 연락사. Okay? For example, one cell and the other cell, okay? For example, in this case, connexin protein can bind to each other, so it can connect each cell. Okay, so connexin for example, in animal cell, connexin. So you have to memorize the protein name. Okay, connexin. Okay, midterm exam. Connexin linked cytoplasm of two cells. Okay, after that, ions and small signal molecule can communicate each other like this. For example, large molecule cannot pass through this gap junction, but small ion or the small signal molecule can pass through the connection. So this is, we have to know gap junction in animal cell and connection. How about the plant cell? Plant cell has this is one plant cell and the other plant cell. This is recorded as plasmodesmata. Okay? So this is recorded as plasmodesmata. So one cell and the other cell. So usually between the cell, this is ER. So this is recorded as decimal tubule. Decimal tubule is also bind to each other. And also, this is plasmodesmata. This is decimal tubule. Between them, we have some space. Okay. So this decimal tubule leaves a tiny gaps. So they have tiny gaps, so through which small metabolites and small ions can pass through like this. Okay. So in the plant, we have to know the terminology. Plasmodesmata. Okay. Plasmodesmata. You can write down 원형질 연락사. Okay. Okay. And final one is we will learn about evolution of multicellular organism. Okay, so usually the one cell can aggregate it, so it will make cluster like this. Also, after they make cluster, each cell can communicate each other. I mentioned that communication is most important. Okay, communication between professor and student is most important, and communication between you guys is also very important. So we have the Marin and Rhea. So you can talk to Marin and Rhea. Where come you from? Okay, which university? Like this. Okay. Also sometimes can talk to each other and also eat lunch together like this. You can communicate each other. So you will know the French culture very easily. Okay? Maybe no one talk to Marin and Rhea. You can talk to each other. Okay? From now on. Okay? Because communication is most important. Okay? After that, some cell become specialized. Okay, some cell, if that specialization of some cells happens, so some cell becomes skin, some cell becomes eye, some cell becomes hair. Okay, some like this. So specialization of some cells, if that organization of specialized cell to become the tissue is like this. Okay, so skin cell becomes the skin. Okay, eye cell becomes eye. Okay, stomach cell becomes stomach, like this. Okay, organization of specialized cell to the tissues, right? Okay. Okay, and tissue becomes organ, organ becomes organ system. Okay, important thing is that one cell becomes, they can aggregate to each other. They become the cluster, and they communicate each other. They can communicate each other. So after they communicate, you know that she is specialized in some part, and she is specialized in some part. He is specialized in some part, like this. Okay? And after that, so specialized cells become, they organize each other, they become tissues like this. One cell become organized like this, they'll become tissues like this. Okay? Okay. So next Monday, we have the midterm exam. Do you have any question in midterm exam? Okay, it's done.